Welcome back to the deep dive. Today, we are putting a really specific, really high-stakes piece of science under the microscope.
We are.
We're looking at a paper from Cell by Rush and colleagues, and it tackles something I think of as the autoimmune paradox.
It's a paradox that, it affects millions of people.
Exactly. So let's just set the scene. Imagine you have a patient with Crohn's disease. Their immune system is confused. It's attacking their own digestive tract.
Right. It sees self as an invader.
So to stop that, we give them these powerful anti-inflammatory drugs. And they work, they stop the attack, but they're basically sledgehammers.
They suppress the entire immune system.
So you might save the gut, but now the patient is suddenly vulnerable to pneumonia or sepsis or even these really nasty fungal infections.
Right, you're turning off the master switch for the whole house when you really just want to dim one light bulb in the living room.
That's a perfect analogy. And the mission of this paper is to find that dimmer switch. Specifically, they're trying to drug a protein called CARD9.
And the way they did it is it's a masterclass in modern drug discovery.
It really is. So we're going to go almost sentence by sentence through their logic, because they took this target that everyone considered, undruggable, and they found a way in.
They absolutely did.
Okay, so before we get to the how, let's define the what. What is CARD9, and why is it the bad guy here?
So CARD9 is a protein, but it's not an enzyme. It doesn't, cut things up or speed up reactions. It's what's called a scaffold protein.
A scaffold. Okay, so it's about structure.
Exactly. It's like a physical hub. It lives inside your myeloid cells, which are your immune system's first responders.
And how does it work? Like, what's the mechanism?
Okay, so think of it like this. On the cell surface, you have a receptor, a little sensor called dectin-1. Its whole job is to sniff out fungus.
Got it.
When dectin-1 bumps into a fungal cell wall, it sends a little ping inside the cell. That ping wakes up CARD9.
And what does CARD9 do when it wakes up?
It builds a team, it grabs two other proteins, BCL10 and MALT1, and they all clump together. They form this huge physical structure. They call it a signalism.
A signalism. It sounds like a transformer or something.
It kind of acts like one. Once this thing forms, it triggers a pathway called NFB.
And that's the fire alarm.
That is the fire alarm. NFB rushes to the nucleus and just screams for the cell to release cytokines inflammatory signals like TNF and IL-6.
So the chain is fungus, dectin-1, CARD9 signalism, NFB, and then massive inflammation.
You got it.
Yeah.
Which is a great system if you actually have a fungal infection. You need that.
Right. But in Crohn's, this pathway is just hyperactive. It's firing all the time,
causing chronic inflammation in the gut.
Okay, so logically, if we just block CARD9, we stop the Crohn's. But here's where I get stuck. If we block it, don't we leave the patient totally open to fungal infections? Aren't we back to the sledgehammer problem?
That is the crucial question. And this is where the paper brings in this huge clue from human genetics. Nature basically ran a clinical trial for us.
The delta 11 mutation.
Exactly. Geneticists found this rare variant in people called CARD9 delta 11. A piece of the protein is just missing. A chunk of the scaffold is gone.
And what happens to people with this mutation?
This is the magic part. These people are protected from Crohn's disease. Their risk drops. But, and this is the key, they do not get severe fungal infections.
And they can still fight off candida.
They can. It's the Goldilocks variant. Not too much inflammation, not too little immunity.
It's just frayed.
It creates what's called a dominant negative effect. The broken protein mixes with the normal protein and just kind of gums up the works enough to lower the inflammation, but not enough to kill the defense system.
So the mission of the paper was to build a drug that perfectly mimics that genetic mutation.
That's the holy grail.
Simple goal. Apparently impossible to execute. Why has no one drugged CARD9 before?
Because CARD9 is a ghost? It's considered undruggable?
I hear that term a lot in pharma. Does that just mean it's really hard, or is it like physically impossible?
It's a geometry problem. Most drugs we have work like a key in a lock. Proteins usually have a deep pocket, an active site where they do their work. You just design a molecule to plug that hole.
But CARD9 is a scaffold.
Right. It's made of these things called coil domains. Imagine two ropes twisted tightly around each other. The surface is smooth and flat. There are no pockets, no grooves.
It's like trying to rock climb up a sheet of glass.
That's a perfect analogy. A standard drug just slides right off. There's nowhere to grab on.
Which brings us to our first recap.
Let's do it.
Okay, so let's lock this in. Our target is CARD9, a scaffold protein driving inflammation. Our inspiration is a genetic mutation, CARD9 delta 11, that protects against Crohn's without wrecking the immune system. And the huge obstacle.
CARD9 is undruggable. It's a smooth protein with no pockets for a drug to bind to.
So how do you drug the undruggable?
You change the rules.
Yeah.
The authors use what they call a binder-first strategy.
A shift in mindset.
A total shift. Usually, you screen for a drug that inhibits the protein. You ask, does this stop the machine?
But here, there's no off switch to look for.
Exactly. So they lowered the bar. They just ask, does anything, literally anything at all, stick to this glass wall?
They just wanted a foothold.
A toehold, a scratch, anything. And to find it, they used a DNA-encoded library, a DEL screen.
Oh, I love these. The scale is just, it's hard to even comprehend.
They screened 22.4 billion unique molecules.
Billion, with a B.
It's like trying to find one specific grain of sand on a beach. They just washed these 22 billion molecules over the CARD9 protein to see if anything would hang on.
And did they find one?
They found a hit. A family of chemicals called benzimidazoles. Let's call the leader compound 1.
Okay, compound 1, it sticks. Does it stop the inflammation?
Nope.
So they found a barnacle.
A functionally silent barnacle. It stuck to one of the coiled domains, but the signal still formed, inflammation still happened. It was a dud, therapeutically.
In a normal drug company, this is where the project dies. It doesn't work, throw it out.
But because this was a binder-first strategy, they saw it as a probe. They used X-ray crystallography to take a picture of where it was sitting on the protein.
And they found something.
They found something the structural biologists had missed. They found the glycine hole.
Okay, the glycine hole. In that smooth rope of a protein, there was this tiny imperfection.
A spot lined with glycine amino acids. And glycine, for anyone who skipped biochem.
It's the smallest amino acid. It's tiny.
The smallest, so it leaves this little gap, this tiny divot in the protein surface. And compound 1 had found that gap.
So the glass wall did have a keyhole. They just had the wrong key.
The key fit in the hole, but it didn't turn the lock. So they moved to phase 2, the displacement screen.
This is king of the hill.
Exactly. They took their silent binder, compound 1, and used it as a placeholder in the hole. Then they threw a new library of chemicals at the protein to see if anything could knock compound 1 out.
They wanted a bully, something that binds even tighter.
And they found one, a new class of chemicals, benzodiazepines.
Wait, benzodiazepines? Like Valium, Xanax? Are we talking about sedating the immune system here?
Chemically similar core structure, but no, these aren't psychoactive. They found this specific molecule, compound 5, which they then refined into compound 6, and this thing kicked the original binder right out.
So this is where it gets really cool. Compound 6 didn't just sit in the hole like the first one did.
No, this is the breakthrough. The first molecule just touched one strand of the protein coil. Compound 6 sat in the glycine hole on one strand, but it was shaped just right so it could reach across and grab the neighboring strand, too.
It's like a staple.
A molecular staple. It locked the two strands together into a rigid, inactive state.
Okay, so we've gone from a barnacle to a staple. How does stapling the protein stop the signaling?
It blocks a really specific enzymatic step. To build that big signalosome, CARD9 needs to be authorized by an enzyme called TRIM62.
Okay.
TRIM62 comes in and attaches a little molecule, ubiquitin, to a specific spot on CARD9. That's the go signal.
The stamp of approval.
Right. But because compound 6 has stapled the protein shut, the shape has changed just enough that TRIM62 physically can't get in there to do its job.
Access denied.
Access denied. So, no ubiquitin stamp means no signalosome assembly, means no NF-keep activation, means no inflammation.
That is just incredibly elegant. But now they have to prove the Goldilocks claim, right? Does it spare the rest of the immune system?
This is the validation phase. They took human immune cells and they gave them two different challenges. Challenge A was zymosin.
Zymosin, which mimics a fungal infection.
Yep. It triggers that dectin-1 receptor, and the result, the drug worked perfectly. Shut down TNF, shut down IL-6, all of it.
Okay, so challenge B.
LPS, which mimics a bacterial infection like E. coli. It triggers a completely different set of receptors.
And a result?
Nothing. The drug had zero effect. The immune system responded to the bacteria with full force.
That's the proof.
That's the proof.
It selectively turns off the fungal, autoimmune pathway, but leaves the bacterial defense totally intact.
They had mimicked the genetics perfectly.
Okay, let's do another quick recap here because we're about to hit the big plot twist.
Right.
We found a silent binder in a hidden glycine hole. We used that to find the stronger molecule, compound 6, of benzodiazepine.
And this new drug acts as a staple, locking CARD9 shut.
which prevents the enzyme TRIM62 from adding that go signal, which kills the inflammation. And crucially, it only works on fungal triggers, not bacterial ones.
Perfect.
So we have a drug that works in human cells. We have the mechanism. Next step, cure the mouse.
And this is where the researchers hit a brick wall.
I feel like every great biology paper has a mouse problem.
It usually does. They gave compound 6 to normal wild-type mice, and it did absolutely nothing.
Zero efficacy.
Zero. The mice were just as inflamed as the mice that got placebo.
So what was wrong? Was the drug breaking down in the blood or something?
Nope, drug was fine. The problem was the target. They went back and looked at the genetic sequence for human CARD9 versus mouse CARD9.
They're usually pretty similar, right?
Very close. But in that one tiny spot, the glycine hole, there was a difference. The human sequence is alanine, glycine, serine.
There's our glycine, the hole maker.
But the mouse sequence is leucine, serine, serine.
So where we have a tiny glycine, they have a larger serine.
And serine has this extra chemical group hanging off it.
It's like someone stuffed a tennis ball in the keyhole.
It completely filled in the hole. The pocket didn't exist in the mouse protein. The drug literally bounced off.
That's a nightmare scenario. You have a drug, works in human cells, but you can't test it in an animal.
And this is often where drugs go to die. But this team, they doubled down. They said, okay, if we can't change the drug to fit the mouse, let's change the mouse.
Enter CRISPR.
Enter CRISPR. They engineered a humanized mouse. They went into the mouse genome and surgically swapped the mouse's LSSA sequence for the human AGSR sequence.
They drilled the hole into the mouse protein.
They created a mouse that is a normal mouse in every single way, except for this one tiny spot on its CARD9 protein, which now has a human-shaped keyhole.
Wow. Okay, so now for the final showdown, the in vivo proof.
This is the last chapter of the paper. They took these humanized mice and they challenged them with a fungal lysate to trigger a massive Crohn's-like inflammatory storm.
And then they treated them with compound 6.
And it worked. The TNF levels in their blood, the main marker for inflammation, dropped significantly. The drug successfully dampened the immune response in a living animal.
And just to prove it wasn't a fluke, they also tried it on the normal mice again.
Yes, the crucial control. In the normal mice, the ones with the blocked pocket, the drug still did nothing.
And that proves it. It's not just some random effect. The drug works exactly by binding to that specific pocket. Pocket open, drug works. Pocket closed, drug fails.
It's a complete validation of the entire strategy.
It really is. It shows this binder-first idea is a real roadmap for drugging the undruggable.
I have to play devil's advocate before we close.
Go for it.
Okay, we are celebrating that we mimicked the genetic mutation, right? We mimicked CARD9 delta 11.
But the people with that mutation who are protected and healthy, they have one copy of it. They're heterozygotes. What about people with 2 copies? Or people with no CARD9 at all?
That's the cautionary tale. Humans with a complete loss of CARD9 are severely immunodeficient. They get these invasive fungal infections in their brain, their spinal cord. It's life-threatening.
And this drug, compound 6, it locks CARD9 in an inactive state. It basically takes it off the board.
It does.
So if I have Crohn's and I take this pill every day for 10 years,
Yeah.
am I mimicking the healthy person with one broken copy? Or is it possible that the drug is so potent I'm actually pushing myself into a state that mimics having no CARD9?
That is the million-dollar clinical question, the therapeutic window. In the mouse study, it was a short burst of treatment. But for IBD, treatment is lifelong.
There's a risk that if you suppress CARD9 too hard for too long, you trade your Crohn's for chronic candidiasis.
Exactly. The paper proves we can hit the target. The next massive challenge is figuring out how to hit it just gently enough to be safe for decades.
Well, even with that question hanging out there, this is just a monumental piece of science. From 22 billion molecules down to a dud to a staple, and proven in amounts they had to build from scratch.
It's a beautiful scientific art.
And a great example of how deep you have to dive to solve these really tricky medical paradoxes. That's a wrap for this episode. Thanks for listening.
Stay curious.